Okay, so welcome back to this next video in which we are discussing the CRISPR-Cas9 system. Okay, right, so we've now discussed how the CRISPR-Cas9 system functions as an adaptive immune system within bacteria. Now specifically, Cas9 is the enzyme which we found in streptococcus pyogenes, and what it stands for is CRISPR-associated protein 9. Okay, now in other bacterial species we have found a huge number of similar proteins which are also known as Cas enzymes for CRISPR-associated protein. Okay, but Cas9 is the specific one from streptococcus pyogenes, and it's incredibly useful, and we use it in genetic engineering. Okay, so let's now discuss how we can use the CRISPR-Cas9 system to generate knockout mice. Okay, so we could produce knockout mice prior to the development of the CRISPR-Cas9 technology. Okay, but it was less good, basically. So what the CRISPR-Cas9 system allows us to do is it allows us to improve our techniques. It's an improvement of the technique. It makes it much easier to generate knockout mice. Okay, so this is about improving genetic engineering technology rather than creating a whole new genetic engineering technology. Okay, right, so let's talk about how we can generate a knockout mouse then. Okay, firstly, let me just tell you a little bit about the genetics of mice. So, mice are diploid just like humans, okay? But they don't have 46 chromosomes. Instead, mice have 40 chromosomes, okay? But just like humans, in the case of humans, it's really 23 pairs of chromosomes. In the case of mice, it's 20 pairs, basically. Okay, right. So let's talk about the early stage of development for a mouse. Okay, so you start off with a fertilized egg cell, and within a few days this has developed into a structure known as a blastocyst. Okay, so let me draw you a picture of what a blastocyst looks like. So a blastocyst consists of an outer layer of cells, okay, which I'll show here, like so. So this outer layer of cells is what's known as the trophectoderm, and the cells within this trophectoderm are known as the trophoblasts. Okay, so I'll draw them here. So a blastocyst overall is a spherical shape, and then the outer surface of the sphere is made up by this outer surface of cells, and these cells in the outer surface of the sphere, they are known as trophoblasts. Okay, so I'm drawing the trophoblasts now. Here are all their nuclei. Okay, so these are trophoblasts. Okay, and the entire layer then is called the trophectoderm, okay, which makes up the surface of this sphere of the blastocyst. Okay, right, so let's colour in the trophoblasts in blue here. Okay, try and colour them completely in blue, like so. Okay, and then I'll just go around once more. Right, so that's the outer layer of a blastocyst. It's made up of these trophoblasts. Now these trophoblasts will go on to generate the extra embryonic tissue. Okay, so these will make the placenta and the extra embryonic membranes. Okay, right, so then you have a cluster of cells that are within the within the blastocyst here. Okay, so I'll divide this mass of cells here into separate cells, okay, like so, and this mass of cells here is known as the inner cell mass. Okay, so that's the name for the entire thing. Now an individual cell in the inner cell mass is then known as an embryoblast. Okay, and this reflects the fact that the entire embryo, okay, is going to actually be made from the cells in the inner cell mass. Okay, so the trophoblasts will generate the extra embryonic tissue. The embryoblasts, which are now being coloured in orange here, those will generate the embryonic tissues. Okay, right. Then you've got a cavity here within the trophectoderm, okay, and this cavity is known as the blastocele. Okay, so here is the blastocele. Right, okay, so that's what a developing mouse, and indeed a developing human, looks like after a few days. Okay, right, so these cells, the cells of the inner cell mass, the embryoblasts, these have another name. They are also known as the embryonic stem cells. Okay, and you can actually extract these embryonic stem cells from a blastocyst and culture them on a plate. Okay, so I can get my plate here, my cell culture plate, and you can actually keep them alive in a cell culture laboratory, basically. Okay, so if you put them in the right conditions, we can actually culture and grow these embryonic stem cells and maintain them alive. Okay, so here is a plate now of embryonic stem cells. Okay, now, just a little bit of nomenclature for you. These embryonic stem cells are what are known as pluripotent embryonic stem cells. And for short, embryonic stem cells are often abbreviated to ESCs. Oh, sorry, it should be pluripotent stem cells. Get rid of the embryonic stem cells. Okay, so pluripotent stem cells. Now, what does the word pluripotent mean? Okay, well, pluripotent means that these stem cells are capable of differentiating into any one of the tissues within the body. Okay, so humans, for instance, have 200 known different cell types within the human body, okay, which are specialized cell types. So, for instance, neurons, red blood cells, hepatocytes, these are all specialized cell types. These embryonic stem cells can differentiate into any one of those, and the word for that is pluripotent. Contrast this now to a bigger word, which is totipotent stem cells. Okay, even the embryonic stem cells are not actually totipotent stem cells. Totipotent means that you can develop into any tissue of the embryo, but also any tissue of the extra embryonic tissue as well. Okay, so remember the trophoblasts here are the cells which can develop into the extra embryonic tissues. The embryoblasts cannot. So neither of these cells that we've got anymore are totipotent stem cells. The fertilized egg cell, okay, and the cells of the morula, if you know about the earlier development prior to the formation of the blastocyst, those were totipotent stem cells, okay. These now are not totipotent stem cells anymore. We've gone down a level and we're now only pluripotent stem cells. Okay, right. So, what you can now do then is you can culture embryonic stem cells, okay? And this is going to be really important in the development of knockout mice, because what we are now going to do is we're going to knock the genes out of these embryonic stem cells, and then we're going to get a blastocyst back again, and we're going to put these embryonic stem cells which have got the gene knocked out back into the blastocyst, back into the inner cell mass, and then we're going to grow that blastocyst on to create a mouse which will have no copies of the gene. Okay, right. It's a little bit more complicated, as we'll see, but we'll come to that. Okay, for now, the first challenge then is how are we actually going to knock the gene of choice out of these embryonic stem cells here? Okay, so let's draw this. Let's say that we have one of our chromosomes here, okay, within one of these mouse embryonic stem cells, okay, and we have some gene that we want to knock out. So let's just call this gene 1, which is the gene that we want to knock out. Okay, or we could call it, let's give it a letter rather than a number. Okay, we'll call it gene A. Okay, and we want to knock gene A out of the mouse genome. Now we have to remember that the mouse genome is diploid. Okay, so the mouse will have another homologous chromosome, okay, which I'll draw here, which will also have a copy of a gene for gene A within it. Okay, now these two genes will not necessarily be utterly identical. They won't have the exact same sequence of organic bases necessarily, but they will both be similar enough that they code for proteins that do the same thing, basically. Okay, so we have these 2 copies of gene A on our 2 homologous chromosomes. So we need to knock them out of the mouse genome. And the way we're going to do this is using CRISPR-Cas9 technology. Okay, so this is the idea. Firstly, well, this is the bit that I'm not going to tell you about, and I'm just going to tell you it's been done. Okay, so firstly, we have developed embryonic stem cells from mice which now have the Cas9 enzyme in them. Okay, so you can buy these cells, basically. You can buy embryonic stem cells from mice which already have had the Cas9 enzyme put into them. Now, mice embryonic stem cells should not have Cas9 within them. Okay, Cas9 comes from a bacterial cell, but we have taken the gene from bacterial cells and we have put it into embryonic stem cells of mice. You can now buy embryonic stem cells which have Cas9 put into them. Okay, so these cells are already full of Cas9, basically. Okay, the idea now is that the experimenter can design mRNAs, okay, which are going to be CRISPR RNAs, okay, which will then bind with a tracer RNA, load onto the Cas9 enzyme, and then they will cut the DNA at places that are complementary to the CRISPR RNA. Okay, so this is the idea. We are trying to knock these genes out. So what we need to do is we need to design CRISPR RNAs which are going to be complementary to a portion of the of the DNA here that is just upstream of the gene. Okay, and we also need to do this for the portion just downstream of the gene. Okay, so this is the idea. We can produce a piece of CRISPR RNA. Okay, and I'll show this here, like so. Okay, so we'll have this portion of the CRISPR RNA is going to be complementary to a strand on this DNA here, in this blue region of the DNA here. Okay, then it will also have that CRISPR repeat region here, okay, which will then bind to the transactivating CRISPR RNA here in yellow. Okay, so what we can do is we can incubate our embryonic stem cells with this piece of CRISPR RNA, this CRRNA, which we have designed, basically, which will have the CRISPR repeat. Firstly, it has to have the CRISPR repeat so that it will bind to the tracer RNA, or the transactivating CRISPR RNA here. Okay, so without that we haven't got anything, basically. Okay, and then we can design this portion here so that it will be complementary to one of the strands within this DNA. Now, we'll say it's going to be complementary to this portion of this strand here. Okay, now if we expose our embryonic stem cells to this CRISPR RNA, then what will happen is the CRISPR RNA will bind to transactivating CRISPR RNAs, and you'll also have transactivating CRISPR RNAs already expressed within the embryonic stem cells. Okay, so the Cas9 and the transactivating CRISPR RNA will already be there. Okay, right, so all you have to do is get the CRISPR RNA into the cells, and then what's going to happen is these complexes are now going to form. Okay, they're going to bind to Cas9 enzymes, like so, and now the Cas9 enzyme is going to find this portion of the DNA here, and then bind to it and produce a double-strand break there. So let's draw this happening. Okay, so here is our DNA here. Okay, then we'll show gene A in red here. Okay, so this is gene A here. Then we've got this portion upstream of gene A in blue here. Okay, and then we've got also this portion downstream of gene A here in vivid purple. Okay, what's now going to happen is our CRISPR RNA is going to bind to the portion that it's complementary to within our DNA here. So this is the CRISPR RNA here. Okay, then we've got the CRISPR repeat in turquoise here, which will then be bound to the transactivating CRISPR RNA in yellow here. Okay, like so. And now, of course, it's brought along with it the Cas9 enzyme here. Okay, and then the Cas9 enzyme will produce a double-strand break in the DNA. Okay, and hence what you'll end up with then now is you'll have your broken end now here in blue. Okay, you will now have cut off this portion here. Okay, so you break the DNA in half, like so. So there's a little portion of the blue portion that flanks gene A left on this second portion. Okay, then we've got gene A here, and then we've got the portion that flanks gene A on the other side. Okay, right. So what we will then do is do the same thing for the other side. So we'll also design a CRISPR RNA that is complementary to this portion here. Okay, and then it will bind up with a transactivating CRISPR RNA. It will load onto a Cas9 enzyme, and then Cas9 will produce a double-strand break here, so that we've now completely chopped out the gene. Okay, right. And you might think, brilliant, that's how we're going to chop out gene A. We just do that now for both strands, okay, for both homologous chromosomes here, and, you know, it'll most likely be the case that the fragments that are upstream and downstream of gene A are going to be very much so analogous to what they will be on the other chromosome. So you might just have to do this twice, basically, and it might work in both cases. Okay, right. And overall what that is going to do is it's going to produce two double-strand breaks that now separate gene A off from the two sides, basically. Okay, right. So here we now have our blue portion, and here is our purple portion, but there's now no gene A separating them. Okay, right. Now, what is the cell's response to this? Well, the cell has ways of repairing this and bringing gene A back in, so we need to do more than just using the CRISPR-Cas9 to cut this hole in this way. Okay, so what is generally also done is that you put in a piece of DNA that is going to act to help join this blue fragment here back to this purple fragment. Okay, and to understand the purpose of this piece of DNA, I need to explain to you the homologous recombination mechanism for DNA repair. Okay, so let's just talk about homologous recombination for DNA repair. Okay, and then we'll see why this is going to be a problem for us trying to make these knockout mice. Okay, right. So, homologous recombination then. Okay, right. So, firstly then, we start off with a double-strand break. Homologous recombination is a mechanism for repairing double-strand breaks, which are abbreviated as DSBs. Okay, so let's start off with our DNA whole, okay, here, and then let's discuss what's going to happen when you make a double-strand break. Okay, so we'll have the double-stranded DNA here in blue. Now, what sort of things can generally make double-strand breaks within DNA? Well, ionizing radiation is capable of producing double-strand breaks in DNA. So things, for instance, like high-frequency electromagnetic radiation, X-rays and gamma rays. In addition, beta particles, which are just flying electrons. Okay, and then also alpha particles, so the nuclei of helium atoms, 2 protons and 2 neutrons, those are also capable of producing double-strand breaks. So let's say that we are getting ionizing radiation hitting our DNA here, and what it's going to do is it's going to smash it up a little bit. Okay, so what you'll end up with is a mess, basically, where you've lost a lot of nucleotides from the middle here, okay, and now they do not overlap. Now it's likely that you'll have overhangs, so you won't just have blunt ends, okay, you'll have these overhangs, and that's because the ionizing radiation has come from one side, so one of these strands is going to get hit harder, basically, than the other strand. Okay, right, so let's colour these portions in blue here. Now, the question is, how are we going to repair this? Well, there are actually two ways that cells have of repairing this. Okay, one is called homologous recombination. The other is called non-homologous end joining. Okay, non-homologous end joining, or NHEJ for short, this is the one that cells actually more commonly use. Okay, and it's quite simple to explain. All you do basically is you get rid of the overhangs, you cut the DNA back, okay, and then you stick the two blunt ends together. That's all non-homologous end joining is. So you lose all of the bit in the middle that has been destroyed by the ionizing radiation. So it does produce mutations, basically. Okay, homologous recombination is rarer to use, but it's better. Okay, it stands a better chance of actually completely returning this back to this, basically. Okay, right. So, what's the principle then of homologous recombination? Well, the principle of a homologous recombination is that if you want to repair this gap, you need to know what nucleotides were actually in that gap. So how are you going to find that information? Well, you can find it, hopefully, from another copy of this chromosome. Okay, now there are two possibilities here. You can either get very, very lucky and use the sister chromatid. Okay, so if you've got a sister chromatid present at the moment. So let me just make sure that we're all on the same page with regards to understanding the difference between homologous chromosomes and sister chromatids. So let's do this in the human rather than in the mouse, where we're more familiar with it. Okay, so basically, if this is a quiescent human cell here, okay, we know that we have 46 chromosomes within the nucleus of every single cell. Okay, so there are 46 chromosomes. However, really it's 23 pairs because we have two chromosome 1s, a maternal chromosome 1 and a paternal chromosome 1. We'll then have two chromosome 2s, a maternal and a paternal 1, and then all the way on to the last autosome, which is chromosome 22, and then you have the sex chromosomes, which are a little bit more complicated. Okay, right, so these two analogous copies of chromosomes that you have, which are not identical but they are very similar, these are known as homologous chromosomes, and all cells of your body except your gametes, okay, your sex cells, will have two copies of each chromosome. They'll have two homologous chromosomes. Now, then the concept of sister chromatids. So, sister chromatids occur in cells which are about to divide. Okay, so if this cell is about to divide, then it needs to copy every single one of its 46 chromosomes, okay, because it's going to now divide into two cells, and each of these daughter cells needs to have a copy of every single one of the 46 chromosomes. So, it therefore has to replicate all of its chromosomes. So every single one of the chromosomes is going to get replicated. So this one here has now been replicated. The other homologous chromosome 1 has been replicated. Both chromosome 2s have been replicated. So let's put some colour on here. Okay, so here are the two homologous chromosome 1s. Here are the two homologous chromosome 1s on this picture. And they have produced identical replicas of each other. Okay, this new chromosome that it has produced, this is known as its sister chromatid. It is absolutely genetically identical to this first one. Okay, so if the cell suffered this double strand break, when it just happens to have a sister chromatid, then it can go to the sister chromatid and say what's supposed to be in this gap, and then it can get that information. Okay? If it's not lucky enough to have a sister chromatid, you can hope and pray that the homologous chromosome has a almost identical region, if not an identical region, which we can use to find out the information that should have been here, basically. So there are those two possibilities of what you can use. You can either use the sister chromatid or the homologous chromosome. Okay, right. So, let's draw this second chromosome, and I'll get another piece of paper now. Okay, right, so let's just draw out what we've already got. So we've got this double strand break here now, okay, which isn't very tidy. It's got these overhangs on each side. Let's put the orientation of each DNA strand on. So this is the 3' end. We can now add a few more little orientations onto here. That's a 5' end. This is a 3' end. This, of course, is 3' because it's anti-parallel. This is 5', this is 3', and this is 5'. Okay, so let's colour these strands in. And now I'm going to colour them in separate colours. Okay, so we'll colour this one in blue here. We'll colour this one in orange here. Okay, we'll colour this one in green here. Okay, and we'll then colour this one in turquoise here. Okay, right, we now have a copy of the chromosome, which is either the homologous chromosome or the sister chromatid, which, let's say, has a region that is completely analogous to this region that has been damaged. Okay, so let's put this here. So this is the region on our other chromosome, which is either a sister chromatid or a homologous chromosome, which is completely, let's say, identical to this portion that is broken here. Okay, so let's show these two strands in separate colours as well. So we'll have this one in red here, okay, and we'll have this one in vivid purple here. Okay, right, so the first thing that is now going to happen then is that we are going to trim these broken ends, basically. Okay, so here we have a free broken end of our DNA, basically. And what we're now going to do is we're going to trim back the strand which has its 5' end facing into the broken end. Okay, so that's this blue strand. This one has its 5' end facing into the broken end. So we're now going to trim that back. So let me show us trimming that back. So it's now become much shorter because we've completely trimmed it back. Okay, so it's only the strand which has its 5' end facing into the broken end, not the strand which has its 3' end facing into the broken end. So here in blue is that strand which has its 5' end facing into the broken end, and now the 3' end completely overhangs, basically. Okay, so we have produced what's known as a 3' overhang here. Okay, we're going to do the same thing for this other strand here. So this 5' strand here is going to be completely trimmed back and will generate a 3' overhang then. Okay, so in green here, here's our 3' overhang. And now in turquoise, this is our strand which has its 5' end facing into the broken end, which has been completely trimmed back. Okay, right. Now what's going to happen is you're going to get a process known as strand invasion occurring. This little fragment here is going to invade our chromosome here. Okay, because remember, we are saying that this original section here is the same as this section here. Okay, this overhang that we have here is therefore the same as a portion here and is complementary to a portion of this red strand here. Okay, so what we're going to do is we're going to tease apart this portion of the red strand and this portion of the purple strand, and we're going to bind this portion of the red strand here to this orange strand, this 3' overhang of the orange strand. Okay, so let's start off by drawing the blue and orange strand here. So here is our orange strand, here is our blue strand, and now what we have done is we've bound that red strand to the 3' overhang of our orange strand here. Okay, and it's also bound to the purple strand still. So we've opened the red and the purple strand up, we've separated them apart, and we've now bound the red strand to that 3' overhang of the orange strand instead, leaving the purple strand now with no complementary strand bound to it anymore at this portion. Okay, now the idea is what you can do is you can extend this orange strand, because remember this is the 3' end of the orange strand. Okay, DNA polymerase can add nucleotides onto the 3' end of a strand. So what it can do is it can create an extension to this orange strand, which is going to be complementary to this red strand. So what you're now going to get is extension. Okay, and by the way, the process of this strand invading into this double-stranded DNA here, this is known as strand invasion. Okay, right. So you're going to now extend the orange strand hugely. So let's show this happening. So here's the blue strand. We're now making the orange strand much larger, basically. Okay, so here it comes. It's now become much larger. Okay, and here is the blue strand still left tiny. Now, as you make the orange strand longer, you're going to be peeling the red strand more and more away from the purple strand. Okay, so you're going to be breaking these two strands apart more and more. Now, that's going to be happening at this side, but at the other side, you are going to be taking the red strand back off the orange strand and putting it back onto the purple strand. Okay, so you're going to be peeling it off this side, putting it back on that side. Okay, because you don't want to completely separate the red and the purple strand apart, so you're going to be replacing the red strand back onto the purple strand after you've finished, basically. So, here is the portion of the red strand that's still bound to the orange strand. This portion has now teased off the orange strand and is bound once again to the purple strand. Okay, so here is the purple strand here. Okay, and here is the red strand here. Okay, right. Now, this is the genius of what we've done. Okay, we've extended this orange strand, okay, hugely, and the hope now is that this is long enough to have a portion that overlaps with this green portion here. So remember, these two fragments, they came from the same portion of DNA. Oops. They came from this original portion of DNA here. Okay? And we have used this piece of DNA, which we're assuming has exactly the same sequence of organic bases as this portion. Okay? So now this orange strand here now has exactly the same sequence of organic bases as it has originally here, and hopefully it's now long enough to overlap with this portion here, okay, which is this portion here. So the idea is that now, here is the blue strand with the orange strand. It's now going to have a portion that overlaps with that light green strand there. Okay, so let's colour this in. So here is the light green strand, here is the turquoise strand, here's the orange strand, which has now been extended hugely, and here is the blue strand. Okay, now what you can do is you can just use DNA polymerase to fill in the gaps, okay, because remember, this is the 3' end of the orange strand, so DNA polymerase can work its way along there and finish that gap there, and then DNA ligase can just bind the orange strand to the turquoise strand. Then on this side, this is the 5' end of this light green strand, this is the 3' end. We can therefore extend this sequence out, and then DNA ligase can fit the gap. Okay, and this will now completely repair our DNA strand, and it now has got the same sequence of organic bases there as what it had previously. So this is how homologous recombination can repair double-strand breaks, basically, and return them to completely what they were before. Okay, so we do not want this to occur. We do not want the cell to just go to the homologous chromosome, which potentially hasn't had its gene cut out yet, and replace this gene with that portion there. Okay, what we want instead to do is put in a piece of DNA that the cell can actually use for homologous recombination, okay, which will allow it to rejoin this blue portion here to this purple portion here with no gene A in the middle, okay. So we're now going to put in a double-stranded fragment of DNA that's going to be used by the cell in homologous recombination to religate this side here with this side here, but without gene A in the middle. And we'll discuss how we're going to do this in the next video.